Hey guys, remember we were talking about bonding and why atoms like to be together? Well, we're going to take that a step further today. And that step further would be macromolecules, the molecules of life. Macro meaning big, so let's take a look. Macromolecules are, there are four major groups. They are polymers, poly meaning many, and they're composed of monomers, mono meaning one. They are formed by condensation or dehydration reactions. And when you look at those words, to condense, if you think of water condensing, the water's being removed from the air. Dehydration, removing water. Let's look at that. So the first type of macromolecule is the carbohydrate. I'm sure you've heard of those. The simplest subunit of a carbohydrate is a monosaccharide, a single sugar. And that monosaccharide means one sugar, literally. You'll notice I'm pointing out these root words. They're very important to help you understand. And the most common type of monosaccharide is glucose. And you can see it pictured here. It's always in a six-sided ring. And that's, you'll notice that the carbons are all in the ring except for the one up top. The next thing we need to consider is disaccharides. When we put two monosaccharides together, it becomes a disaccharide, which literally means two sugars. They form this disaccharide by making a glycosidic linkage. Do you need to know that? You should recognize it. And if you look in the middle, you can see the oxygen that is at the lowest, it's sort of at an angle down in between the two sugars, and this is through dehydration synthesis, which means a water has been lost. You really should know the most common disaccharides. Here is maltose. It is composed of glucose and glucose. Sucrose is composed of glucose and fructose. And if you look at the name fructose and the name sucrose, there's some similarity in their spelling, and that's the way I use to remember it. And sucrose is common table sugar. Lactose, the sugar found in milk, is composed of glucose and galactose. And so that also has a similar name, so it's easy to remember. Let's take a moment now and really look at dehydration synthesis in a little more depth. And if you look at this diagram, you can see that the hydroxyl groups, remember I told you those functional groups would be important, they were circled or squared out for you in red, and they are going to form a bond between these molecules at that point by losing a water molecule. Let's pause for a moment and go to the board. Okay, so in order to better understand dehydration synthesis, I need a little help from my friend here, Miss Larson, my faithful assistant. And if you notice, she and I are both holding tennis balls, okay? These tennis balls are to represent that we are fully bonded. Now remember carbon, and that's what we each are, we're each a sugar molecule composed of carbons. We want to have all of our orbits full of electrons, so that's why we form our bonds. And now, we're two monosaccharides, and we want to form a disaccharide. And in order to form a disaccharide, we have to make a bond. And I want to have you think of making bonds as holding hands. So in order for us to hold hands, we have a little problem, don't we? What's the problem?
Our hands are both full.
Our hands are both full. So what do we have to do so that we can hold hands?
We have to get rid of the tennis ball.
Okay, so if I get rid of this tennis ball, and you get rid of that tennis ball, can we hold hands?
Mm-hmm.
All right, let's try that. We can just drop the tennis balls, and this is going to be our water molecule. Let it go. Bye-bye, water. And now let's form a glycosidic linkage, which actually goes this way. So we see it, just like in the picture. And we've lost one water molecule. There you go, my friends. Okay, so let's look at this in a little more detail again on the screen. So at the top of the screen, you see that we have the two monosaccharides. That would be myself and Miss Larson holding our tennis balls, which are circled in red here. Actually, they're squared in red. And so by one of us losing a hydroxyl group and the other losing a hydrogen, and if you follow the fun red arrows here, you can see that a water molecule is released, and you can see that nice glycosidic linkage where there's an oxygen between two carbons and forming the sucrose molecule at the bottom of your screen. Hydrolysis is another really fun thing that you should know about. And if you look at the word hydrolysis, hydro meaning water and lysis to break, we are breaking a molecule with water. So it's essentially the opposite reaction. So at the top of your screen, you see sucrose with its glycosidic linkage, that oxygen in the middle, and we add a little water and an enzyme, because enzymes make things go faster, and you can see at the bottom of the screen we have successfully separated the two monosaccharides by adding the water molecule. And you can see that the water has split into a hydroxyl group and a hydrogen so that everybody has all their bonding needs fulfilled. The next term that we want to look at is polysaccharides. Poly meaning many, so it literally means many sugars. There's many functions for polysaccharides. One would be storage. And storage is usually in a starch molecule, like in plants. So we have amylose, which is an unbranched version, and amylopectin, which is a branched version. The other type of storage molecule that we see a lot of is called glycogen, and that's how we store sugar in our body so that we can use it later on. Polysaccharides also have a structural function such as cellulose, and that's the stuff that makes plants cell walls so that they're firm. Let's look at those a little more. So here's starch on this screen, and starch has a 1,4 alpha linkage. Now before you get crazy, I want you just to be sort of comfortable with seeing this terminology. You are going to see it. 1 and 4 means which carbons are actually forming the linkage. So it is the number 1 carbon of one of the sugar molecules, and it's making a linkage with the number 4 carbon of the adjacent molecule. The alpha linkage is the orientation of the bond itself. So if you look at this whole long diagram at the bottom, you see that there are 4 sugars that are linked in a line, and you can see that the oxygens are all in the same orientation. There are 2 hydrogens facing up in these glycosidic linkages. This is an alpha linkage. Cellulose, on the other hand, has a 1,4 beta linkage. The beta linkage differs, and if you look at the bottom, at the top they show you the glucose molecules. The bottom they show you the actual polysaccharide. And if you note, please note that the beta linkage, it rotates. So at one point the oxygen is in the upward position, and the next link it's in the like a facing down position. This is the difference between the 2 linkages. Now let's look at starch versus cellulose. They're both composed of glucose. We did notice that. Starch forms a helical molecule when it's all done by bonding, hydrogen bonding, and remember how important we love hydrogen bonding. And the hydroxyl groups are easily available for digestion. We can easily digest starch. Cellulose, on the other hand, because of the beta linkages, forms these parallel bonding with the hydrogen bonds of the molecules, and so it makes it very strong and very difficult to break down. We cannot break down cellulose. Let's look at some diagrams here. So if you look on the left, we have cellulose, and you can see the hydrogen bonds between the long straight molecules. This is what gives it strength, and it does make it difficult for us to digest. We don't have the enzymes to get in there and break that down. Starch, on the right hand side of your screen, you can see it forms like a helix, and all the hydroxyls are on the outside, so it's very easy to break those down, and the hydrogen bonds are in between the helix. Moving right along, let's look at another molecule, very similar components to sugar but very different properties, the lipids. Lipids are composed of carbon, hydrogen, and oxygen, just like our carbohydrates, but they are hydrophobic. That means they are water-fearing. They're afraid of water. They are non-polar. They are also known as fats, phospholipids, and steroids. And we will be seeing these molecules again and again this year. Fats are composed primarily of glycerol, which is a 3-carbon alcohol, and it has 3 fatty acids attached to it. They are attached by an ester linkage, which is formed through, you guessed it, dehydration synthesis. Here is a diagram of a fat. You can see in the green is the glycerol molecule. The ester linkage is highlighted in red for you. You can see that it has a carbon with a double bond O, and then the oxygen is attached to our glycerol molecule. So this was a carboxyl group. If you remember, I told you those functional groups would be important. And if you look at the chains in black, those are just long chains formed of carbon and hydrogen. The one at the bottom is a little different. It has a green double bond. What happens to that? It's not a straight line. It becomes bent. This is the difference between saturated and unsaturated fats. The saturated fats are saturated with hydrogen, so all the carbon's bonds are filled with hydrogen. The unsaturated fats do not have maximum hydrogen content. If you look at the double bond, in order to form that double bond, they had to lose a couple of hydrogens. Phospholipids we'll be seeing in the later on in the cell membrane. They have a polar phosphate head, which allows them to like water, and 2 lipid tails, which don't like water. Steroids, the final type of fat or lipid, have 4 fused rings, and they vary by their functional groups that are attached on the outside. So they all have the 4 rings, and this one you notice has some methane groups on the top right. So that was a lot of information. Next time I see you in class, we'll spend some time working with it. Before then, if you want, you may want to spend a little time going over this video one more time, taking some notes, pausing if you need to. Make full use of it. Use your book, whatever you need to do. After we play with these carbohydrates and fats in class, the next video installment for homework will be on proteins and nucleic acids, the other kinds of macromolecules. Haven't forgotten those. Thought you might need a little break in between. So see you next time.